Good morning, students. In the chapter Principle of Inheritance, now today we do linkage. Before doing continuity with linkage, we just have to know what's the meaning of linkage here. We just do a graph here. That's a chromosome. On a chromosome, that is a gene A. That's a gene A. That is a gene B. Both gene A and gene B are on the same chromosome. It's a homologous chromosome with it. During cell division, we just assume that it is undergoing such kind of thing. And now what will happen here? Gene A remain on the chromosome and gene B goes to the another chromosome. Gene B goes to the another chromosome. We do another possibility here. Gene A here, gene B here, gene B here. During again crossing over, we just assume the crossing over is toward on this arc at this time. So what will happen? Gene B remaining here, gene B remaining here, but gene A goes to the homologous chromosome. Okay, just do another possibility here. There's a gene A, gene B. Another homologous chromosome here. It's another homologous chromosome. Again crossing over. The possibility to transfer gene A to another chromosome is more. Do another thing again. Now, the chances of separation of both of these genes having different character are lesser as compared to these upper thrice examples. When crossing over done here again, then both gene transfer simultaneously. There is no gene on this chromosome. So what we have to see in here in an overall conclusion that we just observe that here gene A and gene B, the distance is so far from both of these genes. Here gene A and gene B, the distance is lesser, but as compared to the first one, we mark it as A. This is B. But crossing over done here also, and both of genes on the same chromosome get separated out. There's a C. Again, in this case, also crossing over done and both of genes on the same chromosome separate out. But in this case, genes are closely related and the distance between two genes are very lesser. That's why they never will get separated in this time. So what we have to do here, We just note down the term linkage. We just say that linkage genes on a chromosome. Genes on a chromosome are known as linked gene. We can say physically association of a gene on a chromosome means they are located on the same chromosome. That's why we can say here they are physically associated here. Linked gene, and the term is called as linkage. Now, when linkage is more, then the chances of separation is lesser. More linkage, lesser be the chance to get separate out. Means when we observe again here in the A section, gene A and gene B, the distance between both of genes are far from each other, but in this case they are very closer to each other, so it is difficult to separate out gene A and B from condition D or D example as compared to example A. We did example B and example C here. Now we are going to talk about the term gene mapping. The term mapping or gene mapping was given by Alfred, and the term linkage or the concept of linkage was given by Thomas Hunt Morgan. T.H. Morgan. That's why he is known as father of experimental genetics. And the father of genetics was the father of genetics was called as Mendel. Mendel is known as father of genetics. What were the consequences that Morgan got the score for the father of experiment? We will talk it later. First of all, gene mapping. Gene mapping was given by Alfred. First of all, he gave the term recombinant frequency, and the second term is gene unit. For example, we are going to say that gene A on a chromosome. Just remember, gene A on a chromosome is 20 unit apart from gene B and 10 unit apart from gene C. So in our first condition is, that is a chromosome here, this point gene A, and this point just assuming gene B. So the distance between two genes, gene A and gene B, is 20 unit apart. That is 20 unit apart. And the distance between gene A and gene C is 10 unit apart, which means there are the two conditions here. If gene C located here toward the right side of A, it is either toward the right side of A or it is either the left side of A. There are two possibilities here for gene C. So we can say, but there is no mention that the distance between gene C and gene B. So we just write it here that the gene C is located toward the right side of gene A and the distance between gene A and gene C is 10 unit apart. So that's a unit in the term. But when we can say that is gene A and gene B, the distance between gene A and gene B is 20%, or the recombinant frequency. The recombinant frequency between gene A and gene B is 20%, and the recombinant frequency between gene A and gene C is a 10%, which is mean that gene A is a 10 unit apart, then the recombinant frequency for the same condition is 10%. And when we can say the gene unit or unit apart is a 20 unit, so we can say the recombinant frequency is 20%, which is means that one more thing we added here, that is we just know more linkage, less to be the chances of get separation, or we can say here more linkage less combination. Less recombination means the chance of separate out between the two genes on a chromosome is lesser, and vice versa. Means if the distance between two genes are more, means recombinant frequency is more, means linkage is less. And when the frequency or recombinant frequency is lesser here, then the chances of separation or get separated between two genes are lesser, and the recombinant frequency is lesser, and linkage is more here. Now linkage group. Linkage group means number of chromosome in a set. Number of chromosome in a set. For example, we are going to talk about human. Human having 23 chromosome. When haploid, only 23 chromosomes, then the linkage group is 23 linkage group. And Drosophila having 4 chromosome in a haploid condition, that is the linkage group of 4 linkage group here. So the The linkage group is depend on the number of the chromosome in a set. For example, human having 23 chromosome in a one set. So human having a 23 linked group, and Drosophila having 4 chromosome in a set. So Drosophila having 4 linked group here. In the next video, we will talk about complete linkage and incomplete linkage.